Good afternoon, everyone. Thank you and welcome to the special Mary Kay Symposium. I'm Dr. Lucy Gilday, the Chief Scientific Officer of Mary Kay. I joined Mary Kay a little over a year ago after 16 years at Procter & Gamble. The opportunity to serve as Mary Kay's Chief Scientific Officer allows me to marry my passion for science and innovation with enriching women's lives. Mary Kay has been a dedicated supporter and contributor to research and recognizes how environmental stressors such as pollution can have a negative impact on skin. As we strive to further understand the impact of pollution, we will also uncover what we can do to intervene and reduce this negative impact. This symposium, in partnership with the Society of Investigative Dermatology, is just the latest example of Mary Kay's ongoing partnerships with academic and scientific communities. We've assembled 4 esteemed speakers for this symposium. You will hear about the dangers of pollution, its impact on skin, how it may already be showing up in the clinic, and some possible strategies to reduce the negative impact of pollution. Without further delay, I would like to introduce our first speaker. Dr. Max Costa is the professor and chairman of the Department of Environmental Medicine at New York University School of Medicine. In addition, he also holds multiple positions that include Professor of Biochemistry and Molecular Pharmacology at New York University School of Medicine, Program Leader, Environmental and Molecular Carcinogenesis at NYU Cancer Center, and Director of Core Center of Excellence in Environmental Health Sciences from NIHS. Dr. Costa has been working in the area of epigenetics and cancer for over 20 years. He has studied how carcinogenic metals affect the epigenetic program through inhibition of histone demethylases. In particular, Dr. Costa has studied how arsenic, nickel, and chromate alter histone post-translational modifications. Dr. Costa has also conducted ChIP-seq using several histone markers and RNA-seq following carcinogenic metal treatment of cells. He has been working on the mechanism of metal carcinogenesis for over 40 years. Dr. Costa has published around 400 peer-reviewed papers in journals of international significance and is a recipient of many prestigious awards, including the Kenneth Morgerege Award for Outstanding Contribution to Toxicology, recipient of a Lifetime Achievement Award from the Metals Specialty section of the Society of Toxicology. Without further delay, I'd like to welcome Dr. Max Costa.
Thank you, Lucy. So I'm going to tell you about air pollution. But just to let you know how important it is, that the WHO, World Health Organization, estimates that air pollution kills 7 million people in the world every year. And about 90% of the inhabitants of the earth, the humans, are breathing what they consider unhealthy air. So this is a really major problem. So, air pollution consists of sulfur oxides, particulate matter, and this is a very important one, both PM 2.5 and PM 10, oxidants such as ozone, carbon monoxide, hydrocarbons, nitrogen oxides, lead, and other heavy metals. So if you look at the world air pollution in terms of PM 2.5, you can see that the United States is pretty clean. But if you look at Eastern Europe and Southeast Asia and China, you see very high levels of PM 2.5 air pollution. In fact, in some of these areas, it reaches 500 micrograms per cubic meter, which is equivalent to smoking 25 cigarettes a day. Now, during the seasons, air pollution varies widely, and if you look in Beijing, during the summer months, air pollution is relatively low, 100 micrograms per cubic meter, but during the autumn and winter months, it goes up to 600 or 700 micrograms per cubic meter, and similarly in New Delhi as well. But focusing more in the United States, so you saw a green map before, but now the United States, if you look more closely, according to the WHO, Southern California, Arizona, Texas, has greater than 10 days where the air pollution is not safe. So by the year 2050, cities like Beijing will have 17,700 mortalities from air pollution, and cities such as New Delhi and Mumbai will have over 50,000 deaths due to air pollution. So air pollution is dependent upon activity in the city. So if the city burns coal, you'll see a lot of aluminum, selenium, cobalt, and arsenic. If you have ferrous and non-ferrous industries, you'll see a lot of zinc, copper, and lead. If you burn oil for power, you'll see a lot of vanadium and nickel. So it's going to vary widely depending upon the activity in the city, and you can correlate where it's coming from by source apportioning the pollutants you measure with the activity in the city. So in terms of the skin, particulate air pollution, which comes from oil burning and other activities, will activate AHR, and reactive oxygen species will activate MAP kinase, ERK, and JUNK, and cause pro-inflammatory cytokines and metalloproteases to be secreted. And using antioxidant drugs or anti-inflammatory material, you can protect against the adverse effects, particularly on the skin, by putting these molecules and preventing the oxidative stress. So we did a lot of studies in Saudi Arabia, studying the air pollution there. One of the studies we did was in Jeddah, Saudi Arabia, where we have a lot of desalination plants, which all their drinking water comes from the ocean, so that they have to desalinate the water. There's oil refineries, and so we studied the air pollution in Jeddah, Saudi Arabia. And what we found was very interesting, that looking at PM2.5, we found that metabolic syndrome was statistically increased due to soil and road dust and traffic. Now, Saudi Arabia has a very high incidence of type 2 diabetes, about 40%. They have a large number of people that are obese, and in fact, some of the air pollution may be contributing to some of these problems. When we took Saudi Arabia air pollution and put it on human bronchial epithelial cells and did RNA-seq, we found that it activated all the genes that were involved in cholesterol and triglyceride synthesis. And when we use U.S. air pollution, this didn't happen. So this is a unique property of the air pollution for Saudi Arabia, which could be influencing the obesity and type 2 diabetes that is occurring there. This is a picture of Sudbury, Canada. So I'm going to talk about nickel, because nickel is a major component of air pollution, particularly if you burn oil. And in Sudbury, Canada, they've been mining nickel there, which, by the way, is only there because a meteor hit there a couple million years ago. All the nickel that's mined in the world comes from outer space, from meteors that hit the Earth. So there's a large meteor that hit the Earth, and they've been mining nickel for a couple hundred years. They used to use open pit smelting, and it killed all the vegetation in the area. So then they built these huge stacks, 1,000 feet high, and now the nickel and sulfur dioxide is blown with the jet stream to New York City. So we can measure periods of very high levels of nickel that are coming from Sudbury as a result of their refining and mining of nickel. So I'm going to tell you about nickel and how it works and what it does to the cell adversely. So the major target for nickel in the cell are the deoxygenase enzymes. These enzymes use iron and alpha-ketoglutarate, and they hydroxylate, so like hydroxylation of collagen, the regulation of HIF-1 alpha, the ALK-B DNA repair enzymes. FTO is a DNA repair enzyme, but polymorphisms in this enzyme are responsible for a lot of obesity in the world. Histone demethylases and TET oncogenes. So the only use of ascorbic acid in the cell is for these enzymes. So this is a typical reaction of a deoxygenase enzyme. This is a histone demethylase. So iron attacks oxygen, and then it attacks the methyl group. Alpha-ketoglutarate is decarboxylated, and you form formaldehyde. So you segmentally demethylate the H3K9 by 2 molecules of formaldehyde. And nickel comes in and easily displaces iron from these enzymes. So the iron is held by 2 histidines in a carboxylic facial triad. Nickel has 3 times the affinity for this active site than iron does, so it easily displaces the iron and completely inhibits these enzymes. So and it does it at micromolar concentrations, so that's the major effect of nickel in the cell. Another important property of these enzymes is the iron that's redox sensitive. So the iron can be oxidized by oxidative stress, hydroxyl radicals, and you need so when the iron gets oxidized to +3, the enzyme becomes inactive. So you need ascorbic acid and glutathione to reduce the iron +3 back to iron +2. So another interesting effect of metals that we've been working on is the ability of arsenic and also nickel to deplete this important protein called stem loop binding protein. So stem loop binding protein is needed to translate canonical histones without a poly-A tail. So nickel and arsenic cause a degradation of stem loop binding protein, and now you cannot get translation of canonical histones. So the default is to add a poly-A tail onto the canonical histones, which do not have a poly-A tail. This causes increased stability of the histones. The canonical histones increase H3, and it produces a lot of disruption of nucleosomes and is carcinogenic. So this is just some data from human B2B cells that have been transfected with the vector alone, or a vector that has H3.1 with a poly-A tail. You can see anchorage-independent growth goes up. If you put the stem loop structure back into the vector, you can suppress the transformation. And the same thing happens with invasion as well. And the mechanism for this seems to be the ability of H3.1 with a poly-A tail to displace non-canonical H3.3, which has a poly-A tail, from the promoter of genes. This is a ChIP-seq experiment where we flag tagged H3.3, and then, so here you see it right in the promoter transcriptional start site. If you transfect H3.1, you displace the 3.3 from hundreds of genes, which decreases their expression. In addition to that, you see a strong mitotic block as a result of polyadenylation of 3.1. 11% of the cells are mitosis versus putting the stem loop. You see H2AX phosphorylation. You see multinucleated cells. You see chromosomal damage. So this really wreaks havoc on the cells to have polyadenylated 3.1. So in summary, air pollution causes 7 million deaths worldwide, and by 2050 will produce more than 50,000 deaths in cities like New Delhi and Mumbai. PM2.5 is one of the most dangerous components of air pollution. Its composition differs greatly depending upon the sources it generated. Studies of PM air pollution in Jeddah have revealed that it is responsible perhaps for metabolic syndromes, hyperglycemia, and upregulation of cholesterol and lipid synthesis genes. Nickel is a major component of PM, and it's a major target in the cell or dioxygenase enzymes. In addition, nickel and arsenic can cause degradation of stem loop binding protein, which results in polyadenylation of canonical histones. This causes genomic instability, carcinogenesis. And one mechanism for this is the ability of polyadenylated H3.1 to displace 3.3 from active gene promoters. Thank you.
Thank you. I think we're going to we'll save some questions towards the end. Okay, great, thank you. So our next esteemed speaker for the day is Dr. Jean Krugman. The title of his talk is Air Pollution and Skin State of the Art. Dr. Krugman is from Minden, Germany, and has obtained his MD from the University of Munster in 1986. After several stays abroad, including international experiences, he became resident assistant associate professor at the University Hospital for Dermatology of Freiburg. From 1994 until 2001, he was full professor, deputy director of the University Hospital for Dermatology, Dusseldorf. In 2001, he was appointed Chair of Environmental Medicine at the Medical Faculty of Heinrich Heine University, Dusseldorf, and thereby also Director of IUF. Under his directorship in 2011, the Institute became a member of the Leibniz Association and was renamed IUF. His research focuses on dermatotoxicology, immunodermatology, and photodermatology, with a special emphasis on environmentally induced skin diseases and skin aging. He is co-author of more than 400 contributions to scientific journals and books, as well as editor of 9 textbooks. Dr. Krupman was awarded several national and international prizes: the Arnold Rickley Award, the Albrecht Fleckenstein Award, Paul Gerson Una Award, Oscar Gans Award, the Series Research Support Award, and the Dermopharmacy Innovation Award. Since 2010, he is a member of the Leopoldina, the German National Academy of Science. Since 2012, he is speaker of the Leibniz Research Alliance Healthy Aging. Additionally, he is distinguished adjunct professor at Fudan University in Shanghai and senior visiting professor of the Human Phenome Institute of Fudan University. He is no stranger to this society. As of yesterday, he received the Tanioko Kihei Memorial Award for his lifetime achievements in dermatology. So without further ado, Dr. Krupman.
Yeah, thank you very much, Lucy, for this very kind introduction, and good afternoon, everybody. I would now like to follow up on the very elegant presentation by Professor Koster and tell you and share with you the existing scientific evidence that air pollution or pollution is also very important when it comes to skin health. You already learned from Max Koster that we can categorize pollutants. This is the classification of the American EPA. And for the sake of this presentation, I will primarily focus on particulate matter, or PM, and I will also briefly touch on nitrogen oxide, because these are the two pollutants where we clearly know that they matter for skin health. And when I say skin health, I actually refer to 2 points, and that is there is now very, very strong scientific evidence that healthy skin is being affected by air pollution, and in fact that air pollution can drive skin aging and in particular skin pigmentation. And this is what I will primarily talk about today, but I would also like to mention, at least here at this point, that there is growing evidence that air pollution can trigger and can aggravate skin diseases, and this is definitely true for eczema, atopic eczema in children, but there are also now more and more reports coming out indicating that eczema in adults and also in the elderly can be worsened by air pollution. There is a clear link to skin cancer, and there is some indirect evidence suggesting that air pollution can also trigger urticaria and possibly acne vulgaris. So let's start with to talk about the link between air pollution and skin pigmentation, skin aging. The first evidence to suggest that there might be a link is this study, which was published in 2010 in the Journal of Investigative Dermatology. And this epidemiological study made use of a cohort which goes by the acronym of SALIA. SALIA stands for the study of the influence of air pollution on lung function, inflammation, and aging in elderly German women. This cohort was started in the mid-80s at that time. Roughly 4,000 women who at that time point were 55 years old were enrolled, and since that time they have been regularly being followed up, and these ladies are of course now 30 more years older, but we are still able to do follow-up investigations. And all these ladies lived in a part of Germany which is called the Ruhr area. And for those of you who are not so familiar with Europe, the Ruhr area is the largest urban agglomeration in Germany. Surface-wise and population-wise, it strongly resembles the London area, and historically it's known for its mining and steel industry. And when you see these pictures, this is how the Ruhr area looked in the 60s and 70s. When I show this to my students, they always think, oh, this must be a northwestern city in China somewhere, but this is how it used to look in the Ruhr area in the 60s and the 70s. So there was a lot of air pollution. And the beauty of the cohort is that we had ladies living in these heavily polluted cities like Duisburg, Gelsenkirchen, Herne, Dortmund. For the soccer fans among you, this is where the best German soccer teams come from. But we also had access to ladies who lived in rural parts where only agriculture was done in Borken and in Dülmen. So we had a very nice gradient as it concerns exposure to air pollutants. We, in one of the follow-up investigations, we then looked at skin aging, and we did this with an instrument which is called Skinexer. Skinexer is a validated score which can be used in field studies very easily and very reliably. And of course, we also carefully measured the extent and the degree of air pollution these ladies have been exposed to, for example, by determining distance of residency to a major road, which is a road with more than 10,000 cars per day. By using GIS, we measured particles from traffic, we determined individual soot exposure and exposure to background concentrations of airborne particles. So the message is we know very, very well the air pollution exposure of these ladies. And when we then looked whether there would be an association between exposure to air pollutants and the manifestation of skin aging traits, we very surprisingly found that indeed there were quite some strong associations, as is indicated here, by odds ratios above 1.2, for example, for soot exposure and the development of facial lentiginous on forehead and cheeks. This is just another way to plot these data, and the message is the higher the soot concentration, the more likely people are developing facial pigment spots. And I should emphasize that, of course, these data have been adjusted for age, for skin sensitivity, for sunburns, use of sunbeds, smoking, and heating with fossil fuels, which are obvious confounders. The observation that there was this very strong association with pigment spots actually prompted us to ask whether we might find similar results also in East Asians, because the development of lentiginous is one of the major clinical hallmarks of skin aging in this ethnicity. And this is indeed the case, and these are results that we have generated in close collaboration with Professor Sijia Wang, who is in the audience, and Professor Li Jin at Fudan University in Shanghai. And here we found that there is also a strong association not only with particles but also with a gas that is with nitrogen oxide. And you can see here that the association between NO2 levels and facial pigment spots, in this case on the cheeks, is very similar also when we compare the German women with the Chinese women. So an increase of 10 micrograms per cubic meter in NO2 was associated with 25% more pigment spots on cheeks in these two groups. So the major conclusion from all of these studies is that chronic exposure to traffic-related air pollution is associated with more facial lentiginous. We have shown this now corroborated and extended in several other studies that we did mainly also with our Chinese colleagues. But I like to say that there are also independent studies done by other investigators which confirm our findings. Nevertheless, all the evidence that there might be a link between air pollution and skin pigmentation is epidemiological in nature, and as you know, epidemiological studies can only describe an association, and this does not automatically mean that this association also makes sense and that there is a cause-effect relationship. And we therefore felt that it will be very important also to provide mechanistic insight into this, and for this purpose, we have recently developed an ex vivo human skin model in which we topically treated human skin with standardized extracts of diesel exhaust particles particles. We did this with very low concentrations. We were able to do this repetitively, thus mimicking chronic exposure to air pollution. And then, of course, because we were interested in skin pigmentation, we first looked at changes in skin color. And you can see this already by bold eye on the left side. You see the models that have been treated with 2 times with diesel exhaust particles or 3 times with diesel exhaust particles. In comparison to untreated models, you see that there is an increase in skin pigmentation, which actually looks a little bit patchy. Of course, this can be measured by chromometry, as is shown on the right side. We then wanted to know whether this is due to an increase in melanin, and the answer was yes. In the upper left part, you see the quantification of total melanin content, which is significantly increased after diesel exhaust particle treatment. We also see it when we stain using Fontana-Masson for melanin on the left side, untreated, on the right side, diesel exhaust particle treated, nice increase in melanin content. And this is also associated with a transcriptional upregulation of many genes, which in one way or the other contribute to melanin de novo synthesis. This is ex vivo, but can we also see this in vivo in normal human skin? And we also recently looked at this by using fin chambers, which allowed us to topically apply the diesel exhaust particles to healthy human skin, again at very low concentrations, repetitively. And again, we found this is decreasing the delta ITER values, or in other words, it's increasing skin pigmentation in some of the individuals. We could take punch biopsies and RT-PCR them, and we found that the gene expression pattern strongly resembles the one that we see in the ex vivo skin model. So now if you take everything together, the epidemiological evidence and the mechanistic evidence, then it is clear that exposure to particulate matter, in particular to diesel exhaust particles, leads to an increase in skin pigmentation. And interestingly, this is not specific for human skin. In September 2017, Science News has entitled "Polluted Water, It's Where Sea Snakes Wear Black." And of course, this raises the question, what is the biological advantage these coral snakes have when they start now to increase the melanin content? And in the corresponding current biology paper, the authors were providing very nice evidence that the increase in skin melanin most likely serves the following purpose. The melanin can bind pollution-associated trace metals, which can then be secreted if the skin is being shed. Whether the same also holds true in humans, whether air pollution is also increasing sloughing in human skin, we currently don't know. We are looking at this, but we also think that there might be an alternative explanation to this, and that is given the fact that eumelanin has antioxidative properties, one could speculate that increase in melanin might serve to limit or reduce pollution-induced oxidative stress responses in human skin. And in keeping with this, we recently found that diesel exhaust depletes antioxidants from the stratum corneum in our in vivo model, and in the ex vivo model we also found that diesel exhaust causes significant lipid peroxidation. And most importantly, when you pre-treat the ex vivo skin models with an antioxidant-containing cosmetic product, then this greatly reduces diesel exhaust particle-induced skin pigmentation. Again, you can see this already with your bold eyes. On the left side and on the right side, you see the corresponding quantification by using chromometry. And with this, I would like to close, and I thank you very much for your kind attention.
Thank you. Just so that everyone knows, after all speakers finish their presentations, we'll invite them on stage and we can ask questions to them towards the end. So thank you for your patience. Our next speaker is Dr. Liang Liu, who has a long-standing research interest in studying genetic and epigenetic mechanisms underlying gene-environment interactions during normal development and disease pathogenesis. He obtained his PhD in developmental biology and molecular genetics from Uppsala University in Sweden, followed by a postdoc fellowship and subsequent research assistant professorship in cancer biology and aging research at the University of Alabama at Birmingham. In 2010, Dr. Liu joined Dr. Cristiano's lab at Columbia University to pursue advanced training in skin biology. He was promoted to assistant professor of dermatology at Columbia University in 2015. His current research focuses on elucidating the molecular pathways underlying the adverse effects of environmental agents such as UV irradiation and air pollution on skin health. Dr. Liu's research has been recognized and funded by American Institute for Cancer Research, the National Institutes of Health, the Dermatology Foundation, and the American Skin Association. The title of his talk today is Antioxidants, a Key Approach to Combat the Effects of Polyaromatic Hydrocarbon Pollution on Human Skin. Dr. Liu?
Thank you, Lucy, for that kind introduction. Okay. And it's truly a great honor to have this opportunity to present our kind of short studies on air pollution and skin health, following these two wonderful talks given by the world leaders in the field of environmental health science. So in the next 20 minutes also, I'm just going to describe to you a topic-specific study that was funded by Mary Kay and in collaboration with the research team at Mary Kay. I wanted to start by sharing this interesting review article that provides a nice summary of some of the most commonly found air pollutants. And the authors also did a nice job in kind of describing those commonly known or suspected health effects on skin. In addition, they provided nice in-depth discussion on the effects of UV radiation and some of the well-known pathways, mechanisms associated with UV-induced skin health effects, which can provide some guidance for our future research on air pollution and skin health. In the past couple of years, our lab at Columbia University have taken advantage of the recent breakthroughs in the multi-omics research field, and we performed different types of high-throughput analysis to identify different types of molecular changes induced by UV radiation, including UV-induced genome-wide gene expression changes, DNA mutation, as well as epigenetic changes. The overall goal is to just using these powerful tools to collect multidimensional biological information that can eventually allow us to perform systems level of analysis to dissect the complex network of events induced by UV to help us to better understand how UV interacts with the skin. So using the large RNA-seq data set from these studies, we were able to perform pathway-focused analysis. As an example shown here that for skin photoaging, we can look at most of the major players involved in this pathway. Our analysis identified both previously described UV target genes as well as new UV target genes that are involved in the extracellular matrix function and formation. For example, our analysis showed that UV can reduce genes involved in ECM production in the meanwhile promoting the activity of extracellular matrix degrading genes. You can imagine that the net outcome would be a loss of ECM in the skin, which is key to the skin firmness and as well as elasticity. And loss of firmness and elasticity of the skin is the hallmark of skin photoaging. Another well-known effect of UV radiation is the induction of oxidative stress by production of excessive amount of reactive oxygen species, which can be visualized using an oxidation-sensitive fluorescent dye as shown in this slide. More importantly, some bioactive compounds such as the antioxidants Narragain, I believe that's the proper way to pronounce it, can dose-dependently inhibit the ROS induction effect by UV radiation. Just provides kind of conceptual or proof of concept in terms of how we can reduce or minimize environmentally induced adverse effects on the skin. So using the genomics data we have, we performed similar pathway analysis to identify genes that are involved, which are shown here as well, summarized here, including both previously described as well as many new genes involved in the ROS production and the metabolism. So following these studies, I'm going through these studies mainly just to provide a kind of a background in terms of how we can use this learned information and apply similar principle towards pollution research. A few months ago, we were able to initiate a research collaboration with Mary Kay to explore some molecular studies on air pollution just to understand the mechanism underlying pollution-induced skin damage. And of course, some of the common pathway and targets we focused on will be similar to what has been done in the UV radiation field. The goal of this short-term, just the recent collaboration with the Mary Kay team is for us to establish in vitro model system or experimental system that allow us first to assess the exposure effects by different pollutants, and also to test bioactive compounds, the efficacy, how they can prevent pollution-induced skin damage. And one of the compounds, or it's a mixture of compounds we used for this experiment, for this project, is provide Mary Kay. It's a mixture of different antioxidants. Our experimental design is fairly straightforward. We used couched primary human keratinocytes. We exposed them to some well-defined pH mixtures or using the diesel exhaust particles as mentioned in Dr. Krutman's talk a moment ago. And then we performed the molecular biology and biochemical studies to assess the effects or the changes induced by the exposure. To test the efficacy of the antioxidant mixture from Mary Kay, we just pre-treated these human keratinocytes with this mixture, then performed similar exposure and subsequent molecular and biochemical analysis for further evaluation. One of the best characterized signaling pathway in the field of pollution research or is the AHR signaling system. So external agents such as pollutants upon entering the cells that can recognize and bind to the AHR receptor and triggers the nuclear translocation of AHR as shown here. Let me see if this one works. Okay. So upon entering the nucleus, the AHR can bind to, recognize and bind to these xenobiotic responsive elements in its target genes to activate its target gene transcription. And CYP1A1 is one of the best known target of AHR, and it encodes an enzyme that is involved in metabolizing the xenobiotics such as the pollutants, DEPs or PAHs. During this process, a large amount of ROS species will be produced that can trigger further downstream cellular events such as production of cytokines or induced DNA damage. The studies shown on this slide provides compelling experimental evidence to support the importance of AHR signaling system in mediating pollution-induced molecular changes in skin cells. The image to the left you can see that upon exposure to BIP, which is one of the PIH chemicals, there's a predominant nuclear translocation of the HVAR protein, which is coupled with a dose-dependent induction of the CYP1A1 gene. For our exposure studies, instead of using any single pH component, we adopted a predefined pH mixture, and the composition is shown to the right of this slide. This composition actually mimics the pH composition in ambient air pollution in the New York City metro area as defined by Dr. Roger Miller's lab a couple of years ago. Dr. Miller's lab also has a custom-made exposure chamber that allow supply aerosolized pollutants into this chamber. Then you can expose your model organisms in the chamber to mimic real-life air pollution exposure. But for our study, we didn't use this chamber. We just added this pH mixture straight into the cell culture. Since no previous studies have looked at this pH mixture on skin or skin cells, we set out to do a dose-response experiment using different concentrations of the pH in the cultured human keratinocytes, and we looked at a bunch of potential target genes as possible target genes based on literature studies. And the results summarized on this slide shows that pH has a consistent, robust induction of the MMP1 gene dose-dependently. At higher doses, however, you do notice, like at 100x, and let me see if I can use this point. I just talked, sorry. You can see at higher doses, you can see a significant growth inhibition as well as toxicity emerged. So for the rest of the pH exposure studies, we settled with 20x concentration, which is well tolerated by these keratinocytes. Shown on this slide also is the effect of pH exposure on HVAR. You can notice that there's little to no effect of pH on HVAR. This is another representative short-term pH exposure experiment we performed, which again showed that pH can significantly induce the expression of MMP1 gene expression. Included in this experiment also is the TAC-SNP gene. pH has a significant induction. TAC-SNP is a well-recognized molecular sensor of cellular oxidative stress that is extensively studied in the field of diabetes research. And upregulation of TAC-SNP by pH indicates pH can also induce cellular oxidative stress in these primary cultured skin cells. More importantly, when we pretreat these primary cultured keratinocytes, you can see a significant reduction of almost complete blockade of these pH-induced molecular changes using these two genes as biomarkers, highlighting the efficacy of this antioxidant blend in preventing skin cells from pH-induced abnormal molecular events. Following these pH exposure studies, we went on to test the DEP exposure, and this composition is based on a formulation that has been used in the Mary Kay lab in the past, and there's also, at this meeting, there's a poster from the Mary Kay lab provide detailed characterization of the physical and biological properties of this composition. As shown on the right of this slide, you can see that DEP can induce robust expression of CYP1A1 gene, consistent with the past reported effects of CYP1A1 induction by pH induction. Also, Pre-treatment with the ATAC or the antioxidant mixture can partially, but not block these molecular changes induced by DEPs. We have also performed biochemical experiments to look at ROS production following DEP exposure. As shown to the left of this slide, you can see that DEP indeed induced ROS production in these cells, which is also blocked by this antioxidant mixture. Looking at gene expression analysis of the ATAC-seq confirmed this effect of molecular changes induced by DEP that is also blocked by ATAC pretreatment. When it comes to the MMP genes, it's interesting to note that the antioxidant alone can reduce the base expression of MMP9 and 10 at basal level, but had little effect on MMP3 gene. Following DEP exposure, however, MMP3 gene is specifically induced, but with little effect on the 9 and 10. Pre-treatment, again, with this antioxidant mixture, almost completely blocked these DEP-induced molecular changes in the cells, confirming the efficacy of this antioxidant blend in preventing these keratinocytes from pollution-induced molecular abnormalities. So after establishing this in vitro experimental system for assessing the exposure effects of specifically defined pollutants, as well as evaluating the efficacy of testing these bioactive compounds, we just went on with a different to explore what other new target genes may be targeted by the pH exposure. So for this purpose, we performed ATAC-seq. The ATAC-seq analysis is based on the knowledge that our genes in the genome are distributed into two different chromatin domains. One portion of the chromatin adopts a more open environment that allows active access of transcription factors to modulate these groups of genes to be transcribed or regulated in response to external stimuli. Another part of our genes are located in the more condensed chromatin areas. And by default, this closed chromatin conformation silence these genes because they can block the access of transcription factors. This method is based on the unique ability of the TNF5 transposase enzyme, which can access open chromatin regions in the genome. It can clips the DNA in the meanwhile, adding a tag sequence tag to the clipped DNA. So this sequence tag serves as the primer for subsequent PCR amplifications and library preparation. These prepared libraries can be sequenced and mapped back to the reference human genome. These mapped sequences represent the genes that are located in the open chromatin areas in the genome that are actively transcribed or regulated. By comparing these different treated and pH non-treated cells, we will obtain the information on which target genes pH can regulate through this chromatin conformation mechanism. So the goal of this experiment is to identify additional new pH target genes. The results of this preliminary ATAC-seq experiment is summarized on this slide. In this experiment, we also included cells exposed to 20 millijoule per centimeter square dose of UV radiation, because our previous, as a positive control, based on our previous studies showing that UV radiation can actively alter chromatin conformation to regulate its target gene activity. As shown on the left graph on this slide, you can see both UV and pH can induce dynamic chromatin changes at over 1,000 genomic sites in the genome. And compared to the UV radiation, pH actually can induce more changes, affects more genomic sites. These genomic sites have been mapped to, or the distribution of these genomic sites are illustrated in the ring maps in the middle. So, as summarized here, pH can, about 50% of the pH target genomic sites are mapped to protein coding regions, whereas 60% of these UVR target genes are mapped to protein coding regions. The advantage of this ATAC-seq method is that it can provide not only information on target genes that encode proteins, it also identify those non-coding genomic regions that may play important gene regulatory functions. On the right of this slide are some of the top-ranked pH and UV target genes, and with those adopting more open chromatin information highlighted in green, where this change to more condensed chromatin information highlighted in pink. So I just wanted to point out that the MMP20 gene is at the top of these top-ranked pH target genes. And no studies so far in the literature has linked pH exposure or pollution exposure and MMP20 activity. Those just opens kind of illustrates the power of this genome-wide analysis in identifying new target genes. I also wanted to point out that the elongation initiation factor 2D, which appears to be a common target of both pH and UVR. These are preliminary analysis. We will perform additional confirmatory validation experiments to validate these findings. In summary, our studies have shown that pH and DEP can activate both MMPs and raw signaling pathway in skin cells. And pH can specifically induce MMP1 expression, whereas the DEP mixture can induce MMP3 gene expression. And we have shown consistently that this antioxidant mixture can effectively block these molecular and biochemical changes induced by these defined pollution components. Also, our ATAC-seq experiment just exemplified the power of using this global discovery approach, these genomics tools to identify new pathways, new targets that can not only help us understand the mechanism, but will also facilitate the development of newer approaches to combat pollution-induced adverse effects on the skin. These are just the conclusions. We have successfully established an in vitro experimental system for both exposure assessment as well as for testing some of these bioactive compounds in preventing pollution-induced effects. And genomic studies will certainly accelerate the identification of new target genes of these common pollutants. Some of the common challenges are summarized in this slide, and it's just due to the complexity of the components of pollution and variation geographically. there's a long way to go really towards achieving a complete understanding how between the interaction of pollution and skin cells. With that, I would like to thank my colleagues at Columbia University, especially Dr. Angela Cristiano, my mentor over the years, who has been extremely supportive to my research efforts. I would also like to thank our Mary Kay collaborators, especially Mary and David, who have already, who not only are very helpful to the scientific aspects of this collaboration, but also really did an excellent job to facilitate everything to make this project possible to conclude within the past 4 months. Okay. And I also would like to thank our past and current funders that have supported my research. And thank you for your time.
Thank you, Dr. Wu. So our final speaker of the special symposium today is really going to bring to bear the molecule to the bedside. So I'm happy to introduce to you Dr. Wendy Roberts. Dr. Roberts is a graduate of Stanford Medical School with 3 postgraduate degrees, which include surgery internship and residency, PGY2, and dermatology residency and dermatopathology fellowship at NYU Medical Center. Dr. Roberts is double board certified in dermatology and dermatopathology. She is founding director of dermatopathology at Loma Linda University Medical Center. She alone pioneered and developed generational dermatology, which is a detailed system of age management. She is also a founder and chair of the Generational Dermatology Symposium, which has become a national meeting venue for dermatologists and plastic surgeons. Dr. Roberts developed the Roberts Skin Type Classification System as seen on the Today Show. Her skin type system is used by doctors around the globe to keep skin of color patients safe while undergoing cosmetic procedures. Dr. Roberts has authored numerous articles and contributed book chapters for the following subjects: acne, chemical peeling, melasma, pigmentation disorders, skin type, aesthetics, skin of color, generational dermatology, geriatric dermatology, sunscreen protection, skin cancer and ethnic skin of color, and pollution of the skin. She is frequently quoted in national magazines, radio, television, and news from around the world. She was cited by Essence magazine as the dermatologist to watch. She was named Rockstar Dermatologist by Glamour magazine and penned the Youth Booster. She is past president of both the Women's Dermatologic Society, which is the 3rd largest dermatology organization in the country, and CalDerm, the California State Dermatology Society. She is also a member of the American Dermatology Association, chair of the geriatric session of the World Congress of Dermatology in Vancouver. She's known by the specialty for her innovation and energy, which I hope you get to see today, and she has been in the private practice in Rancho Mirage, California since 1994. Active in her local medical community, she has been voted as best doctor by our colleagues for the last 12 years, and she was just selected as leading physicians of the world by the prestigious International Association of Healthcare Professionals. So I'd like to welcome Dr. Wendy Roberts, talking today about air pollution, a silent but visible factor in skin pigmentation and photoaging.
Thank you. Thank you. Thank you so much for that incredible introduction, and hello everyone. I really would like to thank you, Dr. Gildea, and Dr. Matsui, and David Gann for inviting me for this prestigious symposium. So I'd like to ask a couple of questions. The first is by show of hands. Who in this room has been to a pollution symposium before? 2 people in the front. Okay, I hadn't either. And my second question is, who here practices clinical dermatology and sees patients? By show of hands. Okay. Well, this is a clinical spin on a very important problem. These are my disclosures. Okay, so for me it all started around 2013, and I started thinking about pigmentation because hyperpigmentation is the #1 diagnosis of skin disease in the world. It really is a unifier on all continents, all types of people, all skin types, all have this #1 problem of hyperpigmentation. And I started to wonder, with many of the readings of our speakers that came before me, what is really going on with hyperpigmentation, and why do we see it so rampantly in some areas? So we know, and we've heard today, that pollution is a major health concern. Dr. Costas gave us some very startling data about deaths. We know that Dr. Krupman talked about the accumulating scientific evidence that air pollution plays an important role in extrinsic aging skin. So, is there a case to be made to consider pollution as a possible emerging etiologic factor, synergistic, and that's the key, synergistic with established risk factors for development of melasma and other facial dyschromias? How can we educate our patients? How can we identify risk factors in patients who don't even know that this invisible etiologic factor is out there? This is our job, to communicate to our patients what they're looking at and what environment their skin is living in. So, looking at these 3 disorders, from left to right, we have melasma in an African American female. We have solar lentigines, otherwise known as Hori's macules, in an Asian female. And in an African female, we have this severe periorbital hyperpigmentation. So what do these all have in common? Well, all of them are increased incidence in skin of color, and all of them have an increased incidence in what I call the global sunbelt, which we'll look at later, and we've actually spoken about a little bit earlier. All of them have an increasing incidence with evolving age. Might environment play a greater role than we think? And could air pollution be another risk factor for the development of melasma, periorbital pigmentation, and solar lentigines? So here we go. Let's look at particulate matter across the world. And we see the red, the dark burgundy, the orange, and the yellow are the highest areas. And this is from a NASA satellite, and we looked at this a little bit earlier in the symposium. But you can really see where these the hot spots of pollution are. This is the pointer. This is the pointer. Is it working?
Battery must have died.
Okay, maybe.
Sorry.
That's okay. Maybe this one. Well, in any case, you can see these areas. And these are actually areas where we see these increased incidents of melasma lentigines. And for those of you who practice clinical dermatology, there are cases that we see, and the pigmentation is a lot deeper, is a lot richer, is a lot more brittle to treat. And we wonder, why is it so hard to clear this pigment? And might it be that, in fact, pollution is a culprit, an invisible culprit? So the sources of air pollution, this I think is most surprising, because global air pollution, we always see pictures of cities and smokestacks and urban, but that's really not the case, because global air pollution is everywhere. It's in volcanoes, like what's happening over in Hawaii, right? Those emissions, lightning, cars, airplanes. You're not even safe in rural areas because there's livestock, there's fertilizer. So these emissions are all over, not just in urban areas, but in rural areas as well. And I think this is really important because we don't kind of focus on that, and when we're speaking with our patients, we have to really take into account the total spectrum, all of the sources of global air pollution. So here are some reasons and effects of air pollution. You know, dermatologists, we like lists of things, right? So this is pretty simplistic, but I just wanted to cover it all. You know, what are the reasons and what are the effects? So carbon dioxide comes from exhaust and energy production. Methane, another culprit, comes from cattle breeding. Sulfur oxides come from exhaust in industries. Fluorocarbons come from refrigerants and propellants. Nitrogen oxides, we heard a lot about that earlier, are from exhaust in industry, and definitely this is a huge culprit in urban areas. Ozone from air with high oxygen levels, soot and particulate from exhaust in industries. The greenhouse effect is also at play here. It keeps the sun warmth and light from reflecting into space and actually condenses pollution, so it's kind of heats up what's already there. Particulate contamination can affect the respiratory system. Raised UV radiation levels can destroy the ozone layer. Acid rain leads to acidification and affects the forest dieback. Increased ozone and then contamination by nitrogen oxides, which affect the respiratory system. So there are a lot of pollutants out there around us all day. And this we kind of went over, so I'm going to skip over this, but there's a lot of evidence-based references, and I just put some of these up, and a lot of accumulative evidence that there is a role for pollution and premature aging of skin and more. Nice review article, 2017, coming out of India, which is great because they have some of the highest levels of pollution and also some of the most stubborn types of facial hyperpigmentation. Dr. Krutman wrote an article looking at a link between acne and pollution. I think this is very interesting, that the observations that Asian women have exacerbations of acne during high pollution periods. Again, clinical information that we need with dealing with our patients. So it doesn't happen overnight, and we talked about generational dermatology, and photo damage occurs over the decades. And this is just a depiction of the decades and what happens. So we really need to advise our patients and institute therapies for our patients so they don't succumb to devastation of hyperpigmentation. And it's just not hyperpigmentation. When you look at the evolving anatomy of photo damage, looking at this Glogau-2 female and a Glogau-4, I'm going to ask you, which skin is healthier? Which barrier is better? Who has the higher transepidermal water loss? Who has the highest corneometer hydration? So when we look at metrics, we know that photo-damaged skin, it's not just cosmetic. Photo-damaged skin does not perform as efficiently and as effectively as normal, young, unphoto-damaged skin. So this is something that is, again, it's not just about aesthetics, it's about skin function, and it's about barrier. So the APMs enter via the nanoparticles, and they can come either transepidermally or enter down the hair follicle, and these nanoparticles generate quinones. what happens is this very vicious cycle of inflammation, of AP1 activation, inflammation, antioxidant degradation, melanocyte activation, and pigmentary changes. And I think we've gone through that. We've talked a lot about the role of antioxidants and actually the ROS that occurs. So here's an H&E of a solar lentigine, and I think the important thing is looking at the dermis. This is a very sun-damaged, photo-damaged dermis, and yes, there are melanotic macules on the cheek, but really what's underlying is just a lot of damaged skin. So how much of this, is it really an active melanocyte, or is it really the dermal changes that are affecting the skin and kind of synergistic with UV and causing this situation? Looking at melasma. Now, melasma has some, MMP activity and has shown some dermal changes with melasma, and there are, vascular variations, and we know now that increased vasculature can also happen in melasma, but it seems like in individuals that these melanocytes are set at a different thermostat. They're sensitive. A person with melasma takes very little sun to hyperpigment. And why is that? Again, when you see the type of deep melasma and extreme darkening of melasma in some of our East Asian, Southeast Asian in particular, and Indian populations, again, we have to think that there is something more at play than just melanocyte hyperactivity. So patient education is very important, and I think that by show of hands, not many of us have been at pollution symposiums, so this is great that Mary Kay is doing this because it heightens our awareness of pollution. And sun protection, facial cleansing, antioxidants, skin lighteners, and dermal regeneration products. These are two patients of mine. They're identical twins, and I love the twin studies because they really teach us so much about our genes plus environment. So identical twins, and you can see the one twin is a smoker, no skincare, kind of natural, right? Doesn't want to do anything. Then the other twin is a non-smoker with skincare, and I think these pictures speak for themselves. Smoking is a type of pollution, and skincare definitely makes a difference. So I like to go through, I'm in that area, when we looked at the map of Southern California, I'm in that area where there's a lot of pollution, and in the United States it really follows the Sun Belt. It's Southern California, Texas, Florida. And that's where you see the increased incidence of melasma and all of these other pigmentary disturbances. So I have patients on a facial cleansing protocol, and it oftentimes is not what you think. I started with the evening. We often start with the morning, but I wanted to go through the evening. So my goal for the evening cleansing is to remove particulate matter and also to regenerate collagen. So I have my patients cleanse their face to remove the particulate matter, and they don't do it with their hands. They should use a cloth or a cleansing brush, or actually a good mechanical cleansing, which will have some exfoliation benefits and really get the face clean. Then I have them apply an antioxidants. I like vitamin C, vitamin E, ferulic. Those are usually my to-go-to antioxidants. And then I also have them use retinol. Tretinoin, generic tretinoin is fine, but, we're talking about dermal regeneration. And we know that tretinoin has been proven to create healthier dermal matrix. So I have them on a retinoid in the evening. And then I have them drink a lot of water, because water indeed is important for, internal cleansing and just removing these pollutants. And that's another point that we don't talk a lot. I think in the past, people have talked about drinking water as kind of this kind of, fluff stuff, esthetician, but it's not. I mean, science has shown dehydrated skin is not as healthy as hydrated skin. So drinking water is really important. And then in the morning, my goal is to create a protective shield for my patients when they go out into the day. So they've already done a deep cleansing in the evening. I don't want to create dry skin, so I have them in the morning just rinse their face with water and then apply an antioxidant. I think everyone should use an SPF 50 or higher. I think the days of using an SPF 25 are gone. I would use an SPF in all skin types, and that's a take-home point. Types 1 through 6, everyone needs sunblock, and I would not differentiate and put, my 4, 5, 6 Fitzpatrick patients on a 25 and put the 1 and 2s on 100. Everyone should be 50 or higher. And then for the natural people out there, I tried to explain that it's not about not being natural. Foundation with sunblock is actually protecting your skin. So I liken it to a car cover. Why do you cover this expensive car? Because you don't want environmental stress on the car. So therefore, you need to cover something that's a lot more precious than that car in your driveway, which is your face, and layer sunblock, antioxidants, foundation, and actually have a shield, because it's not, again, cosmetic. We're talking about health. We're living longer. Our skin has to last longer, so we have to have strategies to protect it. So this is my favorite organization, the Women's Dermatologic Society, and we have an initiative, Play Safe in the Sun, and here we all are out in the sun with our hats and our SPF, and it's a global village, WDS, and we're all lined up here, happy and sun protected. So I've tried to make a case to consider air pollution as another risk factor for melasma, lentiginous, POH, and other disorders. Preliminary evidence, actually, I learned today even more evidence show a positive correlation between extrinsic aging and air pollution. Much work to be done, but I'm confident that with symposiums like this and with increasing research, that our specialty will continue to document the skin effects of air pollution so we can further help our patients, because that, at the end of the day, is what it's all about and why we're all here. And this is my hometown, Rancho Mirage. Thank you.
Thank you, Dr. Roberts. So I'd like to ask all of our esteemed speakers to please join me on stage so we have a few minutes for questions. Anyone who wants to ask a question, please introduce yourself prior to your question. And before closing, I once again want to thank everyone for joining the special Mary Kay Symposium on Pollution and Skin Health. Once again, this is reiterating Mary Kay's commitment to science and academic research. So thank you all for joining us. Great, we see we have a list, a line already. Please introduce yourself.
Hi, first of all, I'm Giuseppe Valacchi from the North Carolina State University. And first I want to thank the sponsor, because I've been in the field of pollution and skin for almost 20 years, and it was very hard a long time ago to make a pollution topic in skin field. So I have several questions, but I don't want to monopolize the discussion, so I just start with one and then let's see what happens. For the first speaker, Dr. Costa, I didn't understand well how nickel can really affect the skin, because I've been working with particulates as well, and I always believed that one of the main compounds that is involved in the radon reaction of particles is iron. It seems that almost nickel makes the iron free, so you have maybe more iron that can do phantom reaction and actually exacerbate the effect.
Yeah, so I don't think the release of iron is that important. It's really in the enzyme itself that has nickel, and those enzymes don't work anymore. So and iron, of course, is very conserved in our body, and of course it's the target for bacterial infection. So iron is really very well regulated in our body. But nickel, the major problem is actually contact dermatitis, which involves the toll-like receptor. So that's a totally different topic,
Yes.
but that's a very big problem with nickel.
Then I have the last question, then I leave this. This maybe is for everybody. So when we talk pollution, is a one word that means a lot of things, as more of you showed. I think this is my comment maybe. I mean, we should underline the fact that not all pollutants are able to penetrate the skin. So when we want to study the mechanism, which I try to do, I think it's good to have an approach where we try to mimic the skin as much as we can, because since some pollutant will never be in touch with the keratinocyte, but maybe the products that they make after interaction with the stratum corneum will be. So just this is a comment that we should make sure that we use the right models when we study pollution.
I fully agree, Giuseppe, and this is why we are using ex vivo skin models.
Yes. So my name is Tarl Pro. I'm from the University of South Australia. And in 2011, we developed a new technology we call microbiopsy, where we can take about 200 cells, about 1 nanogram of RNA, 1 nanogram of DNA from human skin. So we do a lot of studies on non-melanoma skin cancer, UV-induced changes in human skin. The reason I'm here is because I've been approached by a group to look at air pollution effects in skin using this technique. So I have two questions. One, is there any value in minimally invasive monitoring of pollution effects in skin? And then number two, I appreciated the RNA-seq experiment from Dr. Liu, looking for essentially biomarkers that were specific from air pollutants compared to UV, because we've looked at ROS. It's very promiscuous, and when we look with microbiopsy at these things, we want specific biomarkers for either PAH exposure, DEPs, things like this. Do they exist? Or have there been biomarkers that have been validated in humans? I appreciate the in vitro data, but I want to see the human data. So is there any value to minimally invasive monitoring of the effects? And number two, are there human biomarkers?
Yeah.
Thank you.
I will make a brief comment on that. Actually, it's an interesting question. We have done a lot of UV studies, pollution. We just began to do it. So in terms of the biomarker panel, we used our large RNA-seq data set, are still in the process of finalizing the final biomarker panel. We first obtained around 200 UV responsive genes that are consist across different donors in response to different UV doses and time points following exposure. So we take that initial biomarker panel, going through different steps of validation, and we are close to narrowing it down to about 40 genes that still we hope to reduce it further, just in case it makes it to the clinical practice one day, so the smaller will be easier to practice. So yeah, there are human UV biomarkers, but for the pollution biomarkers, I'm not aware, except those gene-specific research in the past that we have just looked at MMP1, MMP genes, the TXNIP, and CYP1A1. So more new genes and the pathways have to be identified to first and also validated. I believe there will be better markers out there. We just don't know yet.
Sounds like we have a lot to do.
Certainly. We'd be happy to talk more about it.
Maybe that's the answer to the first question is yes.
Let me, please allow me to inquire the following. First of all, I thank you for this enlightening talk, but going back to India, Delhi, Bombay, 50,000 deaths annually. From that point of view, we have so much combustion, the associated soot, pollution, coal ash due to new industry. You have mentioned several pollutants, but would you please enlighten me in terms of radioactivity from the uranium compounds? All these components contains a lot of uranium, and due to the combustion, it enhances, it concentrates. It's active, in microgray radiation, if I will look at it, it has enhanced a great deal. That can cause or contribute significantly to the death rate. Plus, there is one thing. It has an enormous significance in immunobiology and the skin. And finally, are there any way to enhance or to engineer, to modify the UV-associated toxicity, particularly if I can use a topical application and engineer the IL-10, how it can be beneficial? I am sorry I have asked several questions, but they are fundamental as I can understand. Would you please enlighten?
Dr. Costa.
So I don't know how much uranium is in India, but uranium is distributed throughout the world if you're near volcanoes, and almost all uranium compounds have some radiation. Like depleted uranium, for example, which is used in the military, has alpha rays, which don't penetrate very far, but if you get exposed to these things, they get into your cell and they're close to the nucleus, the alpha can still damage your DNA and cause mutations and cancer. So, but I'm sorry, I don't know much about uranium.
Professor Costa, it is very important. Let me meet you with you. I know I work on this line. Let us have a discussion.
So what I'd suggest is we talk separately at the end of the symposium on your questions. Okay, thank you. Next question, please.
Question to Dr. Kurtzman. I have two questions regarding your in vivo method. So when you patched the DEP in vivo study, were you able to see actual penetration of a PM getting into the skin? Another question is, did you have any issue getting IRB designing the study?
First question, we never specifically looked for particles, whether they are inside the skin or not, and we're actually not worried about this as long as we have our biological responses. I can't tell you really whether the pigmentation that we see is due to the organic compounds, which are lipophilic and which sit on the particles and can easily penetrate. We see also a lot of AH receptor activation, which is clearly indicative that some of the organic compounds penetrate. But we also have some other evidence that the particles can penetrate because some of the, I didn't show you the omics data that we have, but some of the gene clusters that we see being induced cannot be explained by an AH receptor response, but are very reminiscent of what we see in vitro, what the particles can do. Yeah, I hope this answers your question.
So I actually have a question for Dr. Roberts. So what's great about the symposium is we get to talk about the advancements in science when it comes to this topic. I'm curious, in your practice, are you seeing an increase in awareness from consumers or concerns that they're communicating in the clinic with respect to pollution and its impact on skin? What are you seeing in your practice?
Well, you know, I think the media has gotten onto pollution and, you know, the beauty editors and magazines, but for the everyday woman and man in the street, no.
Right.
I think people are completely ignorant, really, about it and don't take it into effect. The younger demographic, yes, more than the older demographic. But if you're not really hooked into those, you know, kind of media, trendy type of things, then you're really not.
Right.
The everyday person, my everyday patient, you know, in the Palm Springs area, no. They are completely unaware of pollution.
Right. So in
And
your
how-
practice, do you start to communicate the impacts of pollution as well as other factors on skin?
Oh, yes, always. I
Great.
think, you know, that's my job to, you know, bring people up to date.
Right.
And science and what's evolving, and we know now pollution does affect aging. So it's important for, you know, all the generations, you know, younger and older, to protect themselves from the skin, as I was saying
Right.
earlier about, you know, being natural. You want to be smart
Right.
and be responsive to the changing environment.
Great. Great. Thank you. We have time for one or two more questions. One, yes?
Yes. Hi. Thank you for the fascinating lectures. And I have a specific question for Dr. Liu. So you're talking about UV responsive gene and thinking about to make gene markers for UV responsive genes. So I'm wondering, because through the talk in the conference, we know different ethnicity or skin of color responds differently. So have you thought about your gene markers is going to be really across the skin of color spectrum, or more focused on one particular color?
That's an excellent point and question, really. In our experimental design for this project, we did consider the large variations in terms of not only skin type, but also like individual background, as well as like UV conditions, follow-up time points. So we have considered our different variations, and we try to include samples that can represent or address these variations. That's why we are in a continuous process to kind of trashing the genes that were initially included in our first panel. So I think it's a long process, but we are getting closer.
Well, we are out of time, and I once again want to thank everyone for joining the Mary Kay Special Symposium, and I especially want to thank all of our esteemed panel of speakers for their time and sharing the great information on the advancements of pollution and skin health. So thank you all. Have a great rest of your symposium.